Bacteria grow on solid media as colonies. A colony is defined as a visible mass of microorganisms originating from a single mother cell. Key features of these bacterial colonies serve as an important criteria for their identification. Colony morphology is the visual culture characteristics of a bacterial colony on an agar plate. The visual observation of colony morphology is a valuable skill in the microbiology laboratory for identifying microorganisms. So, colonies must be well separated from other colonies in order to observe the distinctive shape, size, color, surface appearance, and texture. Characterization of bacterial culture in agar plates. So, what is an agar plate? Agar plates are sterile petri dishes filled with agar solidified growth medium that are used to culture bacteria, fungi, or other microorganisms. Selective compounds are sometimes added to influence growth. Therefore, agar plates are the standard solid support material for growing microorganisms. The cultural characteristics are helpful in the identification of a certain bacteria species. These are the color, which describes the pigmentation of the colonies. Surface, it is the appearance of the surface of a colony. Like, how does the surface of the colony appear? Is it smooth, rough, wrinkled, or dull? Shape, it is the basic shape of a colony. The size of the colony can be a useful characteristic for identification. The diameter of a representative colony may be measured in millimeters. The margin is the edge of a colony, while the elevation describes the side view of a colony. The odor is the smell of the bacterial culture, and the opacity describes its transparency. Some of the pigmentation of the colony includes black, it can be brown, light yellow or milky, red or pink, white, orange, opaque or transparent. When it comes to surface characteristics, it can be dry or powdery, smooth or glistening, wrinkled or rugose, mucoid, contoured, radiate, and concentric. In terms of the shape of a colony, it can be punctiform, circular, filamentous, rhizoid, spindle, and irregular. The size of a colony can be pinpoint, pinhead, and matchhead. In terms of margin of colony, it can be entire, undulate, arrows or serrate, filamentous, curled, and lobate. In terms of the colony's elevation, it can be flat, raised, umbinate, convex, crateriform, or pulvinate. The odor of a colony may be absent or it may resemble to something. you should note the color of the surrounding medium with regards to the imparts pigment to surrounding medium. Now let's jump into bacterial colonial morphology with different cultural characteristics. So the following photos includes the aforementioned characteristics of a bacterial culture in agar plates. So the first photo has a brown pigment, irregular, and is translucent. This one here is quite obvious. It has an orange pigment, round, entire, and it is opaque. Here now is irregular. It has a buff pigment. It is undulate and opaque. This one here has a yellow pigment. It is punctiform, round in shape, and is opaque. This one has a yellow pigment, also round, entire, and is also opaque. So these has the characteristics of being round, entire, opaque, and is a large colonies. Here now is filamentous. It is irregular and is opaque. The next one is a small colonies. It has a red pigment. It is round, entire, and it is opaque. Now this one is large. Its color is creamy white and it is circular in shape. So this one right here looks like the egg yolk. So it is circular, glistening, and is yellow. Now this one right here has an irregular form. It is glistening and it is white. Now this one looks like a sponge. So it is yellow, it is veined and rough, and it is rhizoid in shape. Now this one is filamentous. It is rough in surface and is white in color. The next one is, it is entire. This is a lobate. And this one has a margin of curled. Now this is the undulate margin. This is what it looks like. And this is the Characterization. Now this number one right here is raised. Its elevation is raised and is yellow. The number two right here, its elevation is flat and it is a cream colony in front of yellow colony. This one is umbonate. And this one right here is a convex elevation.